to the American Fisheries Society Continuing Education course on infectious diseases of salmonid fish. The video is divided into sections on basic methods, bacteriology, virology, and parasitology. In each section, you will see demonstrations of procedures which have been described in the course text, either as part of a chapter or as an appendix. This video is intended to familiarize you with the basic diagnostic techniques and methods for identification of different types of infectious agents. The methods I will describe are commonly used, but each laboratory will have developed their own protocols. So, if you are collecting samples for someone else, you should consult them for specific instructions. Necropsy procedures for basic examinations. The first consideration in sampling is to determine the number of fish to examine. In fish health inspections for certification, routine monitoring, and pre-release exams, you are often trying to detect a pathogen in the absence of disease. For these exams, the sample size is generally based on an assumed prevalence of infection of 5%. For example, to detect a pathogen with a confidence of 95% in a pond of 500 fish, you need to examine 55 fish. However, when a disease outbreak is occurring, the pathogen will be present in a larger number of fish and fewer need to be examined to determine the cause. During an epizootic, collect 5 or more live fish showing clinical disease signs. Dead fish should not be used unless absolutely necessary because post-mortem changes and the rapid growth of opportunistic bacteria will confuse the diagnosis. The second consideration is the type of sample to take. A general examination for presence of any pathogen will be much different than an exam to detect a specific pathogen. Methods for detecting bacterial, viral, and parasitic pathogens differ in the tissues sampled and the necessity for sterile conditions. If samples are to be collected for assay using immunological methods like the ELISA or molecular methods such as the PCR, specific collection protocols will be required and the laboratory conducting the test should be consulted. A third consideration is the size of the fish. Necropsy procedures for fry are necessarily different than procedures for adult fish. For example, when sampling fry for virus, the entire fish is processed rather than a specific tissue. In this section, a basic necropsy procedure will be demonstrated. After the samples are collected, they will be processed and examined in future chapters on bacterial, viral, and parasitic detection methods. Whether the necropsy is done in the field or the laboratory, a clean working area is required. In the field, this may simply be a board or table with clean paper towels. Supplies should be readily available, and a suitable necropsy kit would contain a variety of forceps, scissors, scalpels, and blades, small butcher's knife, alcohol and a burner for sterilizing tools, iodophor or other disinfectants for cleaning the work surface, paper towels, glass microscope slides and cover slips, fixatives, bacteriological media, containers for samples, pencils and pens for labeling, and ice or refrigeration if samples need to remain cold for transport. In addition, you will need a compound microscope with at least a low and medium power objective for examining fresh samples. The necropsy procedure begins by euthanizing the fish. For small fish, reduce the quantity of water in the container to a minimum, then add a fish anesthetic such as tricaine methane sulfonate, or MS-222, at 1 milligram per mil. Euthanasia of unconscious fish can be completed by severing the spine just behind the cranium with a scalpel. Large fish can be killed by a sharp blow to the cranium, by severing the spine with a knife, or by overdose with anesthetic. The fish should be laid on the work surface with its abdomen facing you so that the body cavity can be easily opened and examined. Before opening the fish, first examine the body surface. Protruding eyeballs are common to many diseases, and the condition is referred to as exophthalmia or pop-eye. Some of the abnormalities to note in the gills are pale color, excess mucus, and erosion of the gill filaments. To examine the gill, lift or remove the operculum, Then carefully cut out all or a portion of a gill arch. This is done by grasping the arch by the cartilage with forceps and cutting it out using a scalpel or small scissors. Lay the gill in a drop of water on a clean microscope slide. If the gill is large, remove the cartilage with a scalpel, then gently lay a cover slip over the gill filaments and add water if necessary. On the fins, look for fraying, excess mucus, fungus, and hemorrhaging, and encysted parasites which may be visible as white or black spots. A portion of the fin may be clipped off and placed on a slide with water and a cover slip for closer examination. On the skin surface, the mucus layer should be thin and evenly distributed. To examine for external parasites, gently scrape a small amount of mucus from the skin behind the pectoral fins and the base of the tail, and also from any lesions. Smear the mucus onto a clean microscope slide, adding a drop of water and a cover slip. Samples taken from the gills and body surface should be examined immediately under a microscope by scanning first at low power, or 100x total magnification, and again at medium power. Use of the high-power oil immersion objective is usually impractical because of the thickness of the samples, but bacterial infections can often be detected at lower magnifications by looking for bacterial colonies in areas with pathology. First, disinfect the body surface with 70% alcohol or iodine and open the body cavity using either sterile scissors or a scalpel flamed in alcohol. Make an incision above the pectoral fin and cut along the swim bladder to the vent. The body wall may be held back or completely removed. While cutting, be careful not to damage or rupture any of the internal organs. First, examine the internal organs and the musculature. The digestive tract includes the esophagus, which extends from the oral cavity to the stomach, the muscular stomach, and the intestine. The pyloric ceca are finger-like outpockets of the digestive tract and are surrounded by adipose tissue and pancreatic cells. Abnormalities that should be noted include fluids, excess mucus in the intestine, and hemorrhages. The liver lies in front of the stomach and is connected to the digestive tract by the gallbladder and the bile duct. It is normally a uniform reddish-brown, so make a note if the color is pale, dark, or blotchy. Hematopoietic or blood-forming organs include the kidney and spleen, and in certain infections these tissues may be enlarged and discolored. Samples for bacteriological and viral examinations are taken immediately after the fish is opened to minimize contamination. Unless your lab is fully equipped with incubators and diagnostic reagents, you will likely be collecting these samples for examination elsewhere. Bacteriological samples are routinely taken from the kidney. If lesions are apparent in other tissues, samples should be taken from these also using the same procedures. To sample the kidney, either cut out or pull the intestinal tissues away from the swim bladder, then gently strip the swim bladder from the kidney surface. To inoculate a sample onto bacteriological media, flame a metal loop until red hot, then insert it into the anterior part of the kidney. The sample is immediately inoculated onto a plate of sterile agar by streaking back and forth across the surface of the agar at one end of the plate. This is termed the primary inoculum. To streak for isolation, reflame the loop, allow it to cool, and make several streaks across the primary inoculum into a new area of the plate. This is repeated 3 to 4 times to ensure the bacteria is diluted over the surface of the agar. In addition to inoculating media, make a smear from the kidney and lesions to stain and examine for bacteria. Using a flamed inoculating loop, go back into the kidney tissue and smear a thin layer of material onto a clean microscope slide. If the smear is thick, a drop of water may be added to spread the cells into a single layer. This tissue is allowed to air dry and then passed quickly over a flame to fix the cells to the slide. The procedures for staining and examining these smears will be discussed later in the discussion on bacteriology. If samples are to be analyzed using a technique such as the ELISA or PCR, you may be required to aseptically excise a portion of kidney and place it in a sample tube. Specific instructions for the technique and the amount of tissue to collect will be determined by the examining laboratory. Samples for viral isolation are collected next. Depending on the examination protocol, tissues from more than one fish may be pooled. Because these samples will be treated to inhibit bacterial and fungal growth, adherence to strict sterile technique is not required. However, it is important to sterilize tools between fish or between groups of fish to avoid cross-contamination of samples. The tissues normally taken are the kidney, spleen, and the gill filaments. Using a scalpel, cut out a portion of the mid-kidney about 1/2 centimeter squared. This is done by cutting along both edges of the kidney, then making perpendicular cuts. If the spleen is less than 1 cubic centimeter, it can be removed whole. If larger, a portion can be excised. The gill filaments should be cut from the gill arches with scissors. All of these samples are placed in a labeled container and refrigerated or held on ice until transport to the examining lab. There are a few other things to consider when collecting samples. Prior to collecting any sample, contact the pathology laboratory to discuss what samples are appropriate and when they will arrive. If fish are to be transported for examination, the best method is to transport them live on ice. An alternative is to transport freshly killed fish on ice. Transport should be as quickly as possible, but never longer than 48 hours. In addition to the routine samples collected in a necropsy examination, it is often useful to obtain tissues and blood samples. Tissues collected for histological examination can be used to look for abnormalities at the organ or the cellular level and to study the pathology of an infection. Blood samples are useful for a variety of analyses, including examination for pathogens, measurement of hematocrits or packed cell volumes, and determination of antibody titers. Samples for histological examination should be collected as soon after the fish is killed as possible because tissues undergo autolysis rapidly. Tissues commonly examined are the gill, liver, kidney, and spleen, although most tissues are of diagnostic value. Portions of tissue should be cut out using a sharp scalpel to minimize compression, and the thickness of the sample should not exceed 1/2 centimeter. Very small fry may be preserved whole by making cuts in the muscle behind the cranium and in the abdominal wall to ensure the fixative has access to all tissues. Tissue should be placed immediately in a volume of fixative that is at least 10 times, but preferably 30 times that of the sample. For example, a cubic centimeter sized piece of tissue should be placed in at least 10 mils of fixative. A variety of fixatives are used, but the most common is 10% phosphate buffered formalin. All containers should be labeled clearly and the relevant data also recorded on a separate sheet. For transport, make sure container tops are secure, wrapped in absorbent material, and packed into at least 2 watertight bags. Send data sheets either separately or outside the bags containing the samples. Whole blood or plasma is collected using capillary tubes or syringes containing the anti-clotting agent heparin. A capillary tube is commonly used for determination of hematocrits, making a blood smear on a slide, or collecting blood from a small fish. To collect blood in a capillary tube, the easiest method is to cut the caudal tail completely off between the anal fin and the base of the caudal fin, then hold the tube against the dorsal aorta until filled. Place a drop of the blood on a slide, then seal the tube. To make a blood smear, Place a second slide at an angle to the first until it just touches the drop of blood, then push it forward across the slide to make a thin film. Let the smear air dry and stain with an appropriate stain such as Leishman Giemsa. Larger amounts of blood can be collected by several methods, but puncture of the dorsal aorta is perhaps the easiest, and the fish does not have to be killed for this procedure. Use an 18-gauge needle for large fish, a 21-gauge needle for small fish, and a syringe that has been rinsed with heparin. Insert the needle at the midline between the anal fin and the caudal peduncle until you feel the backbone. Apply slight back pressure and move the needle slightly until the blood begins to flow. When an adequate sample is collected, withdraw the syringe, remove the needle, and expel the blood into a tube that can be spun in a centrifuge. Depending on the analysis to be performed, whole blood may be stored up to several hours on ice. Plasma can be prepared by centrifuging the blood, drawing the fluid off of the cell pellet, and freezing the fluid. The blood cells can then be discarded. This concludes the basic methods chapter of this video. If you have any questions, you may wish to review the video and consult your text before continuing. The methods described in this chapter will be used in later chapters, and it is important that you understand how they are performed. In the second chapter, we will cover how to diagnose infections caused by bacterial pathogens. Some of the basic methods you learned in the first chapter will be reviewed here, and new techniques will be introduced. Without a fully equipped laboratory, it will be necessary to send samples elsewhere for definitive diagnosis. However, basic characterization of the organism can usually be done if culture media, basic supplies and staining reagents, a microscope, and an autoclave are available. An 18-degree incubator is helpful, but a clean bench top in a room with a stable temperature of 18 to 22 degrees will suffice for growing most bacteria. The first consideration in culturing bacteria is the choice of media. Some bacteria are very fastidious and require special nutrients. However, tryptic soy agar and brain-heart infusion agar are commonly used for routine isolation as they support the growth of most commonly encountered salmonid pathogens. In addition to this general media, it is common to inoculate tryptic yeast extract or cytophaga agar if infection by a member of the Flavobacterium group is suspected. Media must be prepared aseptically, and agar plates should be stored in the refrigerator stacked in canisters or plastic bags with the agar surface uppermost to prevent pooling of condensed water. Sampling and inoculation of media. Fish should be alive when collected or not dead for longer than 30 minutes because contaminating bacteria grow quickly. As described during the general necropsy examination, samples for bacteriology are generally taken from the kidney and from internal and external lesions. To culture bacteria from a skin or gill lesion, do not disinfect the lesion. Flame the loop until red hot and insert it into the leading edge of the lesion. By going into the lesion with a hot loop, you will kill off some of the surface bacteria, which may be largely contaminants. Inoculate the agar by streaking back and forth gently near one edge of the plate. Reflame the loop, let it cool, and make several streaks through the primary inoculum into a new region of the plate. Repeat this several times, each time picking up less of the inoculum so that the bacteria will be diluted to form individual colonies. Label the bottom of the plate and incubate with the agar surface on top, either in an incubator or on a bench top. In addition to inoculating the sample onto media, you may make a smear to stain. Reflame the loop and insert it into the tissue. Make a thin smear of the tissue on a clean labeled microscope slide. The slide can be air-dried, then heat-fixed for storage. Samples collected in the field that cannot be inoculated immediately onto media can be stored on ice for up to 24 hours. The sample may consist of a portion of tissue which has been excised aseptically or taken using a plastic loop or sterile swab. To do the latter, first puncture the kidney membrane with a sterile scalpel and then insert the loop or swab. The swab is then placed in a labeled sterile tube and refrigerated or held on ice until it can be inoculated onto agar. Bacterial colonies will normally be visible within 48 to 72 hours of inoculation. The shape and pigmentation of the colonies should be noted. Varied types of colonies and those which do not grow on the streak lines usually indicate contaminants. Identification of the bacteria. Performing a gram stain allows differentiating of bacteria into two groups, those that stain blue-black or gram-positive, those that stain red are gram-negative. Many common bacterial fish pathogens are short rods that stain gram-negative, although several important pathogens, such as the one that causes bacterial kidney disease, are gram-positive. To determine the gram staining characteristics for the bacteria you have isolated, flame a loop and carefully pick a portion of a single bacterial colony from the plate. Mix the bacteria in a drop of water on a clean slide. Allow the smear to air dry and heat fix it by passing it over a flame. A gram stain may also be done directly on a smear from a lesion. First, flood the slide with crystal violet dye for 30 to 60 seconds. Drain the slide and rinse with tap water, then flood with iodine for 30 seconds. Drain and rinse the slide again, then carefully decolorize with an acetone alcohol solution by adding 1 drop at a time until the solution runs clear. Rinse to stop decolorization and counterstain with safranin for 30 seconds. After a final rinse, gently blot the slide and allow it to air dry. Finally, place a drop of microscope immersion oil on the stained smear and examine using the 100x oil objective. Gram-positive cells will appear blue to purple and gram-negative cells are red. If some cells are red and others blue, this may mean a mixed culture of bacteria, incomplete decolorizing, or that the culture was old. When examining for gram staining characteristics, also note the shape of the bacteria, as this is an additional differentiating trait. Another test that is easy to conduct and provides important information is a motility test. There are several methods to determine if bacteria are motile, but the easiest is to make a wet mount from a fresh culture. Use a sterile loop to mix a small amount of bacteria in a drop of water on a slide and carefully apply a cover slip. Bacteria can be examined using the 40x objective. Motile bacteria will be actively swimming. Non-motile bacteria will not have any directional movement but will appear to vibrate randomly. Presumptive identification of bacteria is usually made by performing a variety of biochemical tests which follow a dichotomous key as outlined in the Fish Health Section Blue Book. Reagents can be prepared from recipes in basic bacteriology manuals or can be purchased. These tests detect enzymes and metabolic products characteristic for the bacteria and determine what carbohydrates the bacteria can utilize. The test for cytochrome oxidase is one of the simplest of these tests and is useful for differentiating between gram-negative bacteria. Oxidase test strips are commercially available, and the procedure simply involves smearing bacteria from a single colony onto the shaded area of the test strip and looking for a purple color change within 20 seconds. Use either a sterile toothpick or a plastic loop because iron loops can give a false positive reaction. Other biochemical tests are performed either by streaking or stabbing bacteria from isolated colonies into tubes of media or wells in an identification system. Results are generally obtained after 24 hours when the bacteria have had sufficient time to grow and react with the media. Most tests have a dye incorporated so that the results are read as a color reaction. Antibiotic sensitivity testing can be done as part of the identification procedure. For example, sensitivity to Vibrio Stat 0129 identifies the bacterium as a Vibrio species. However, a more common reason for testing antibiotic sensitivity is to determine what treatment will be most effective. In this assay, a culture grown from a single isolated colony is spread onto a fresh agar plate to create a confluent bacterial lawn. Then discs impregnated with antibiotic are placed onto the agar. The bacteria are allowed to grow for 24 to 48 hours, then observed for the circular zones of inhibition surrounding the antibiotic disc. The larger the clear circle, the more sensitive the bacteria is to the antibiotic. The protocol described here is a quick method for determining the relative sensitivity, but standardized and more quantitative protocols are becoming more commonly used. Confirmatory identification of most bacterial fish pathogens is based on serological tests. These tests include the fluorescent antibody technique, agglutination assays, and the enzyme-linked immunosorbent assay, or ELISA. Each of these techniques uses antibody prepared against the pathogen of interest. The fluorescent antibody test, or FAT, can be used to identify bacteria from smears of tissues and fluids, or from histological sections, as well as from culture. The test can be conducted as a direct test, or DFAT, with the antibody against the bacteria conjugated with a fluorescing dye, or as an indirect test, or IFAT, where a secondary conjugated antibody is used. To perform a direct FAT, prepare a smear of bacteria from an isolated colony, or use a smear taken from the kidney or a lesion. Allow the smear to air dry and fix with heat or methanol. An appropriate positive control would be a known sample of the suspected bacteria. For a negative control, use another species of bacteria or tissue from an uninfected control fish. The fluorochrome conjugated antibody should be diluted according to the manufacturer's instructions. Place the slide in a dark, humid chamber and add a drop of conjugate to the smear. Allow the antibody to react for at least 5 minutes, but up to 1 hour, depending on the test recommendations. Gently rinse the antibody off the slide by squirting phosphate-buffered saline onto the slide above the smear and letting it run down over the smear. Then flood the slide with PBS for at least 2 minutes. Briefly counterstain the smear with Evans Blue, rinse again with PBS, and air dry. Immediately add buffered glycerol mounting fluid, apply a cover slip, and examine using a microscope equipped for fluorescence microscopy. If the fluorochrome fluorescein is used, positive bacteria will appear apple green when examined under oil immersion with a 100x objective. An indirect FAT is conducted similarly, but the antibody prepared against the bacteria is unconjugated. Therefore, this procedure requires a second step using a conjugated secondary antibody. Agglutination tests are routinely performed on pure cultures of bacteria. There are several forms of agglutination test, some of which can be obtained commercially. The simplest of these is the whole cell agglutination test. In this assay, bacteria from an isolated colony are emulsified in a drop of saline on a microscope slide, and a drop of specific anisera is added. For a negative control, add a drop of saline to a similar suspension of bacteria. For a positive control, add the anisera to a known suspension of the suspected bacteria. Gently mix the bacteria and anisera by rocking the slide for 2 to 3 minutes. A positive reaction is indicated by clumping of the cells, which can be seen using the naked eye by holding the slide over a dark background or by viewing with the microscope at low magnification. The ELISA procedure usually requires that tissues be collected and frozen or processed immediately to preserve antigens. The ELISA is an extremely sensitive technique, and as with the other serological procedures, there are many variations. In general, specific antisera is absorbed onto the wells of a 96-well plate. Tissue collected from the fish is emulsified to release the bacterial antigens, and a sample of this is added to the well and incubated. After unbound antigen and tissue are removed by washing, an antibody enzyme conjugate is added. Unbound antibody is removed by washing, and an enzyme substrate is added to give a color reaction which denotes a positive sample. Details of this procedure will differ according to the specific assay, so it is best to consult the laboratory that will be performing the test. There are some commercial tests available that simplify the procedure by providing pre-coated plates or tubes, pre-diluted reagents, and simple tissue preparation methods. Of the numerous bacterial pathogens that affect salmon and trout, relatively few are isolated routinely. The bacteria that cause bacterial gill disease, cold water disease, and columnaris disease are all gram-negative long thin rods. Bacterial gill disease is associated with stresses like crowding and poor environmental conditions. As the name implies, the gills are primarily affected, and signs of disease include flared opercles, fused gill filaments, and lethargic behavior. Cold water disease usually occurs when temperatures are below 12 degrees centigrade. Signs of infection typically include skin and muscle lesions that occur first on the peduncle area and can be quite severe, causing erosion of the skin and muscle and loss of the caudal fin. If isolated on Psittophaga agar, the yellow raised colonies are distinctive. Columnaris disease usually occurs when temperatures are greater than 14 degrees centigrade and environmental stressors are present. Disease signs include ulcerated lesions on the skin and gills that appear pale to yellowish. On Psittophaga agar, this bacterium forms dry rhizoid yellowish colonies. Furonculosis, enteric redmouth, and vibriosis are septicemic diseases caused by gram-negative bacteria. Signs of acute septicemic diseases may be very similar, and diagnosis should rely on the history of disease at that location and isolation of the causative agent. Furonculosis is named for the distinctive skin lesions or furuncles that may occur, especially in chronic disease conditions. However, in acute infections, death usually occurs before clinical disease signs. The bacterium is easily recognized upon isolation because it is pigmented, and colonies in agar turn a deep brown color. Enteric redmouth takes its name from the hemorrhaging or reddening that may be apparent in the mouth. The bacterium is motile and has distinct biochemical characteristics. Vibriosis is a disease that occurs in the marine environment. Disease signs may include hemorrhaging, visible at the base of the fins, the vent, and around the mouth. The bacteria can be isolated on general media and identified by their morphology and biochemical characteristics and their sensitivity to Vibriostat O129. Picericatsiosis is a disease that typically occurs in net pen culture and is currently not widespread. However, because this pathogen has caused severe problems in other regions, it is of regulatory importance. Disease signs are varied, but if present, lesions of the liver are distinctive. Although the organism can be detected from cells in the kidney by staining, the bacterium will not grow on agar media and must be cultured on cells similar to a virus. There are only a few diseases caused by gram-positive pathogens. The most common of these is bacterial kidney disease. Bacterial kidney disease is typically chronic in nature and is characterized by gray lesions in the kidney. This bacterium does not grow well on typical agar and must be cultivated on media containing special supplements. In addition to being fastidious, the bacteria is slow-growing and may take several weeks to produce visible colonies. Therefore, diagnosis is often done using an alternate procedure like the FAT or the ELISA. Although these bacteria are isolated most commonly or are of most concern, other bacteria can also cause severe disease and high mortality. Before treating for any bacterial disease, it is important to obtain a definitive diagnosis. This chapter covers collection and handling of samples for virus assay. Because most methods for virus identification require having facilities for maintaining fish cell lines, those techniques will only be introduced here. Samples for virus assay are routinely collected from adult fish and from fry, but may be taken from any size fish, particularly if a loss is occurring. However, the choice of tissue varies with the age and life stage of the fish. For fry, the entire fish is sampled, removing the yolk sac if it is still present. For fish between 4 and 6 centimeters, the entire viscera, including the kidney, is removed. For larger juvenile fish, portions of the gill, kidney, pyloric ceca, and spleen may be collected. If testing adult salmon, the sex fluids are generally collected at spawning. When spawning the female, a portion of the eggs and fluid are expressed into a clean cup and at least 1 ml of the ovarian fluid is collected. An easy method is to draw the fluid into a 3 to 5 cc syringe without a needle. The syringe cap is left on to exclude drawing up eggs. From the male, approximately 0.5 milliliter of milk may be collected directly into a clean cup and placed into a small test tube. If the adult fish are lethally spawned, samples of kidney, spleen, pyloric ceca, and gill filament no larger than 1/2 square centimeter or about the size of a pencil eraser are also collected. Samples should be collected as cleanly as possible, and dissecting tools should be sterilized to prevent cross-contamination between fish. Convenient sample containers may include screw or snap cap tubes and Ziploc bags. Fish can be sampled individually, combining the organ samples for each fish into a single container. Depending on the number of fish sampled and the purpose for examination, samples from several fish may be pooled. When pooling samples, tissues from no more than 5 fish should be combined, and the size of tissues should be consistent between fish. Tissues and fluids should be refrigerated or held on ice, but must not be frozen. Samples should be transported to the diagnostic laboratory within 48 hours. Before tissue samples can be inoculated onto the cell cultures, the tissues must be homogenized to release the viral particles and decontaminated to prevent bacterial and fungal growth. Tissue samples are weighed, then diluted in a buffered salt solution before homogenizing. After thoroughly dispersing the tissue in the media, cellular material is removed by centrifugation. The supernatant, which contains the viral particles, is transferred to a clean tube, and antibiotics and antifungals are added. Milk samples do not need to be homogenized, but cellular material should be removed by centrifugation. Milk and ovarian fluids are decontaminated by filtration or dilution in antibiotics. When performing a general viral screening, 1 or 2 cell lines with a broad sensitivity should be used. Examples include epithelioma papillosum cyprini, Chinook salmon embryo, and rainbow trout gonad cell lines. When screening for a specific virus, a cell line sensitive to that agent should be selected. Cell cultures should be inoculated when they are 80 to 90% confluent monolayers and not older than 48 hours. At least 50 microliters of the sample should be inoculated per 1 square centimeter of cell monolayer. Cell cultures are incubated at 15 degrees centigrade in culture media. Cells are observed for cytopathological changes, or CPE, at 14 to 21 days post-inoculation. When CPE is detected, cells are subcultured or analyzed using a confirmatory test. Although a presumptive identification can often be made on the basis of the disease history of the stock, the clinical signs, and the CPE, a confirmatory test is necessary for definitive identification. One of the most widely accepted confirmatory assays is a serum neutralization test. In this assay, a dilution of the virus isolated from the sample is incubated with an antibody against a specific presumptive virus. It is then inoculated back onto fresh cells and observed for development of cytopathic effect. If the antibody is specific for the virus will be neutralized and a significant reduction of CPE will be observed. However, this method is time-consuming, and other procedures like the fluorescent antibody technique, the ELISA, and the polymerase chain reaction are more rapid and becoming routine. Although more than 20 viruses have been reported from species of salmon and trout, only a few are commonly encountered and cause major problems in fish culture. Infectious hematopoietic necrosis virus, or IHNV, causes an acute septicemic infection in fry. In larger fish, disease is less common and usually chronic in nature. Upon isolation, the virus displays a CPE described as grape-like clusters of round refractal cells on the CHSE214 cell line and as plaques on the EPC cell line. Infectious pancreatic necrosis virus, or IPNV, is another virus that primarily affects fry. Signs of the disease are similar to that of other viral infections and include dark pigmentation, exophthalmia, abdominal distension, and hemorrhaging. When isolated on the RTG2 cell line, the cells round and become spindle-shaped. Two viruses that have appeared recently in North America are viral hemorrhagic septicemia virus and infectious salmon anemia virus. These are currently of high regulatory priority, but their impacts are as yet unknown. Erythrocytic necrosis virus and erythrocytic inclusion body virus are two viral pathogens that infect blood cells, causing anemia in the fish. These viruses will not grow in standard cell lines, so diagnosis is based on observation of typical inclusions in blood smears. A blood smear is made as described under basic methods. After the smear dries, it is fixed in 100% methanol for 5 minutes, then stained with pinocyanol chloride for 2 minutes. Cells are examined under oil using the 100x objective for characteristic inclusions. Other stains may be used as outlined in the Fish Health Blue Book. Because viruses cannot be treated with antibiotics, good culture practices, regular monitoring, and avoidance are critical for minimizing losses due to viral pathogens. This final chapter covers general procedures used for diagnosis of parasites and fungal infections. There are hundreds of protozoan and metazoan parasites identified from salmonids, and this chapter will only provide an overview. For more information, refer to the Blue Book or to references like Parasites of North American Freshwater Fishes by Hoffman and Protozoan Parasites of Fishes by Lohm and Dakova. Parasites occur naturally in fish populations, but may cause problems when water quality is poor or if fish become stressed or overcrowded. Common signs of parasitic infections include scraping against objects and flashing, excess mucus production, and fish may go off feed and become lethargic. Gasping for air at the water surface or flaring of the operculum may occur if gills are heavily infected. Parasites associated with disease are diverse, but can generally be grouped as follows. Protozoa are single-celled organisms generally detected using a microscope. Ciliated, amoeboid, sporozoan, and flagellated protozoa are commonly found on the gills and skin, although some species are found internally in the blood and intestine. Microsporidia are microscopic spore-forming parasites that live inside the host cell. Although they are characterized by their polar filaments, these are not easily seen under the microscope, and presumptive diagnosis is often based on their location and whether or not they form large cysts in the infected tissue. Myxozoans are also microscopic, but are actually multicellular organisms with complex life cycles that require an aquatic worm as an alternate host. There are many species, and they are characterized by the spores they form in fish. Most appear to cause little problem even in heavy infections. However, a few species, like Myxobolus cerebralis, Ceratomyxa shasta, and the PKX organism, can be extremely pathogenic. There are a variety of parasitic worms found commonly on fish, and they can cause problems if infections are heavy or if they infect a critical organ. Monogenetic flatworms are small and use hooks for attachment. These species have simple life cycles and complete their development on the fish, usually on the gills or body surface. Digenetic trematodes or flukes use suckers for attachment and may occur in fish either as adults in the digestive tract or as larval forms called metacercaria, which penetrate tissue. Digenetic species have complex life cycles involving several hosts, which may include snails, birds, and mammals. Cestodes or tapeworms have a flattened, segmented body and a head with suckers or hooks for attachment. Tapeworms have a complex life cycle as well and may occur as adults in the fish's intestine or as metacestodes in the muscle or internal organs of the fish. Nematodes, or roundworms, are thin, cylindrical worms with a rigid body wall. These worms also have complex life cycles. Fish can be host for either the adult form, which is usually found in the intestinal tract, or the larval stages, which infect tissues like the liver and muscle. Acanthocephala, or spiny-headed worms, may be round or flattened, but are characterized by the elaborate proboscis, which bears hooks for attachment. They have complex life cycles with larvae developing in the body of planktonic crustacea. Fish become infected by eating these crustacea, and the adult worm matures in the fish's intestine. Leeches are often found on the skin and gills, and their segmented bodies have suckers for attachment. Although they rarely cause severe problems, they can serve as vectors for other pathogens. Crustaceans not only act as intermediate hosts for fish parasites, but may be parasitic themselves. Parasitic copepods having the appearance of lice, worms, or grubs attached to the gills, fins, and skin of the fish. In large numbers, they may cause serious problems both in culture and in the wild. Larvae of most freshwater clams go through a parasitic stage on the fins and gills of fish. These glochidia encapsulate and may resemble cysts of trematode metacercaria. However, under the microscope, they appear as miniature versions of adult clams. Proper identification of most fish parasites depends on measurements and characteristics of live specimens. Therefore, it is preferable to examine live fish. If this is not possible, fish should be freshly killed and packed on ice. However, following death of the host, certain parasites may exhibit morphological changes or may migrate from their site of infection, and external parasites may detach from the dead host. Freezing is not recommended. However, frozen samples are acceptable for diagnosis of certain spore-forming parasites. If examination of fresh samples is not practical, it is generally sufficient to fix the sample or tissue in 5 to 10% neutral buffered formalin for 24 hours, then to transfer the samples to 70% ethanol for shipping or storage. However, for positive identification of certain parasites, it may be necessary to preserve fresh specimens according to a specific protocol. For example, identification of parasitic worms may require relaxation of the parasite in hot fixative or clearing for identification of hard parts. In these cases, it is best to consult a diagnostic laboratory for instructions. A parasitological examination can be done using the general necropsy techniques demonstrated in the first chapter. Remember, if examination is concurrent with collection of samples for bacteriology and virology, samples for bacterial isolation are taken prior to examining for internal parasites. To detect blood parasites, collect samples immediately after the fish is killed and before the blood clots. Blood should be collected as described in the first chapter using a heparinized container, or the fish may be opened and a few drops of blood removed from the heart. To detect flagellates, dilute a drop of blood in phosphate buffered saline on a microscope slide, apply a cover slip, and examine immediately. Blood flagellates are motile and will be visible using the 10 to 20x objective. A blood smear should also be prepared and fixed in methanol for later staining and examination. Stages of certain myxozoan parasites may be detected in wet mounds or stained smears. Most external parasites can be detected in mucus smears taken from a few sites along the body, including the head, along the lateral line, and the fins. Mucus should be gently scraped using the non-cutting edge of a scalpel and mixed with PBS on a slide. Sample from any area of discoloration or where scales are raised or sloughed. This method can also be used to collect samples from the nares and the oral cavity. Apply a cover slip and examine the sample at 10 to 40x using a compound microscope. There are a variety of parasites that can be found on the skin surface, and many of these only cause problems when present in large numbers. Some protozoa, like Ichthyobodo, Trichodina, and Chilodonella are motile, while others like Trichophora and Epistylus attach to the fish directly or by a stalk. The protozoan that causes ick, Ichthyopterius multifilus, may be visible to the naked eye as tiny white spots peppering the body's surface, and under magnification it appears as a ciliated stage with a horseshoe-shaped nucleus. Areas where scales are raised by underlying cysts or appear as red sores may indicate presence of the myxozoan Myxobolus squamalis. In a scraping from these cysts, the characteristic spores of this parasite are round with two polar capsules. Gyrodactylus species are common monogenean parasites of salmonids, and the hooks they use for attachment may cause direct physical damage of the skin or gill epithelium. Several digenean trematodes insist in the skin and may be visible as black dots. To detect parasites associated with the gills, first examine the opercular cavity for macroscopic parasites, then remove the opercular flap, place it in saline in a petri dish, and examine using a dissecting microscope. To examine the gills in a small fish, remove the entire gill arch. For larger fish, remove several filaments. Place the portions of gill on a microscope slide in a drop of PBS and examine for parasites using the low-power objectives of a compound microscope. Parasites found on the skin are often found in the opercular cavity as well. Terms like fish louse, sea louse, and gill maggot refer to the various species of parasitic copepods that are found on fish in fresh and seawater. These macroscopic parasites are easily detected on the gills, skin, and fins. Cysts in gill filaments that are visible to the naked eye or under low magnification may be indicative of infection by microsporidia like Loma salmoni, myxozoans like Hennegaia, or of the fungal-like Dermocystidium. Eyes should be grossly examined for abnormalities. For closer examination, gently remove the eye using forceps and scissors, placing saline in a petri dish, and examine using a dissecting microscope. The eye can be dissected and the lens, humor, and retina examined using the dissecting or the compound microscope. Certain species of parasitic worms, including the eye fluke, may be found in the eye, and infections may result in blindness. After the fish has been opened, the viscera can be removed by cutting the esophagus at the anterior end and the intestine at the vent. The swim bladder is carefully removed, exposing the kidney, which can now be excised. Each organ should be placed in a petri dish and covered with PBS, then examined for cysts and other visible parasites using a dissecting microscope. These can then be placed on a microscope slide and examined more closely at low magnification. It is common, especially in wild fish, to find a variety of parasitic worms. These typically do not cause disease unless infections are heavy. However, they can cause lesions in the fish, destroying muscle quality, and a few can infect humans. After any visible parasites are removed, the intestinal tract should be stretched out and cut open longitudinally. Again, remove any visible parasites. If the fish is small, examine the entire intestine using the dissecting microscope. If large, make a scraping of the intestinal contents. Protozoan parasites common in the intestine include hexametid ciliates, which may cause problems if present in large numbers. The myxozoan Ceratomyxa shasta also infects the intestine and may be visible as a kidney bean-shaped spore. The intact gallbladder can be placed directly on a microscope slide and teased apart to examine as a wet mount, or if large enough, the fluid can be extracted using a needle and syringe and examined directly. Parasitic worms may insist in the walls of the bladder, and myxozoan parasites are often present in the fluid. After thoroughly examining the digestive tract, examine the other organs. Squashes and imprints can be prepared from the kidney, spleen, and gonads and examined under the 10 to 40x objectives. Metacercaria of the trematode Nanophytes salmonella often insist in the kidney, muscle, or in the eye. This parasite, common in the Pacific Northwest, has been called the salmon poisoning fluke because it hosts a rickettsia that can be lethal to dogs that ingest raw salmon. If kidneys are grossly enlarged, it may indicate proliferative kidney disease. To examine for the PKX parasite, prepare an imprint of the tissue by excising a small portion, blotting off excess fluid, then gently impressing on a microscope slide. Let the smear dry, then stain with Giemsa stain. Look for round, lightly staining parasites that have one to several secondary cells within a primary cell. Another parasite that can be detected in imprints from the kidney or spleen is the microsporidian Nucleospora salmonis. This organism will be evident as an oval spore in the nuclei of developing blood cells. Thin slices of muscle can be cut and examined either by placing the muscle on a slide and holding up to a light source to detect parasite cysts, or by squashing the tissue and examining microscopically. The myxozoan parasites Myxobolus insidiosus and species of Kudoa can be detected in this manner. The myxozoan Myxobolus cerebralis is unusual because it infects the cartilage tissue, causing spinal and cranial deformities in fish that survive infection. Procedures for its identification can be quite labor-intensive, as diagnosis relies on identification of the parasite spores either in histological sections or in digestive cartilage tissue. The latter method is described in detail in the Blue Book, but because examination for this parasite is often required for certification, it will be described here briefly. First, heads from the fish to be tested are cut in half, retaining one half for histology. Next, deflesh the heads by heating in a water bath. All bone and cartilage is then placed in a solution containing the enzyme pepsin, which is incubated for several hours at 37 degrees centigrade. Then the undigested material is transferred to a trypsin solution for further digestion at room temperature. When the cartilage is mostly digested, pour the material through cheesecloth and centrifuge the filtrate to pellet the spores. The pellet is resuspended in a small volume of buffered formalin and examined for spores using the 40X objective. Identification of the round spores, approximately 8 to 10 microns in diameter, is usually confirmed by histology using the other half of the head. During examination for parasites, fungal infections may also be detected. These are often visible to the naked eye on external surfaces and gills and appear as fuzzy growths or as whitish masses, as seen in this saprolegnia infection. However, some fungi affect internal organs or the blood system, and the infections are not as apparent and will only be detected by examination for fungal hyphae in wet mounds or stained preparations. Ichthyophonous infections can be diagnosed by detection of the pleomorphic or variably sized resting spores, especially in smears or imprints of kidney tissue. Procedures exist for isolating and culturing fungi on artificial media like cornmeal agar or Sabaron's agar. However, this should only be done from live or freshly killed fish to prevent isolation of saprophytic or opportunistic fungi. The most effective method of controlling parasitic as well as other pathogens is avoidance. Hatcheries can be managed to reduce the potential for parasites to cause problems by maintaining water supplies free of fish and other aquatic organisms, avoiding transfer of fish between hatcheries, removing dead and sick fish regularly, and reducing stress by stocking at optimal densities and minimizing handling. Infestations can also be controlled using disinfection procedures that may include draining and drying ponds and raceways and disinfecting equipment. Certain external parasites and fungi can be killed using treatments of formalin, potassium permanganate, or hydrogen peroxide, or by manipulating water temperature. For internal protozoan and metazoan parasites, treatments are more limited and must be prescribed by a qualified veterinarian. Before considering any treatment, a fish health professional should be consulted.
When I see rainbow jumping, make me want to pinch my barb, 'cause she's so fine. Yes, when I see rainbow jumping, Lord, make me want to pinch my barb, 'cause she's so fine. But when I see white dog popping in a patch of scum, honey, I don't even want to wet my line. Rainbow gonna leap against the line, honey. Don't you know your brown trout gonna dig? Yes, your rainbow gonna leap against the line, sweetheart. Don't you know your brown trout gonna dig? I bet your white dog gonna get down on all fours and wallow, honey, sort of like you snagged a passing pig. Brook trout look like jewelry, diamonds and rubies shine like stars. Yes, your brook trout look like jewelry, diamonds and rubies shine like stars. I bet your white dog look like something sold only on television, honey, that you'd use to vacuum out your car.